We are live now.
So we can begin then.
Yes, Dr. Katoch, please start.
Thank you, Dr. Saab. Thank you very much.
One request to the
Well, it's my pleasure to.
Dr. Katoch, I was just making one request. If you can request all the participants to mute
Here this afternoon session.
I think he's gone offline. Some problem is there.
I'll just say one very few sentence about she's a highly decorated person, but very, very incisive, I think the medical virologist actually, who has contributed in depth to really deal with the diseases like rotavirus infections, other infections. But when this COVID came, I think from the very beginning, she was in the forefront of thinking process. Independent thinking, I think imaginary and a lot of imaginative. And today we are very privileged to listen to her, on a subject which I think is very, very controversial in the sense that people are even in double mind what they should do. This option or that option or third option. So I think in that context, Professor Gagandeep, I think you will be the best person to deal with the subject because I said the correct your personality, your sharpness, your contributions, your experience, expertise, and then your independent mind. I think that gives it, I think, a lot of value to your personality and to us as a listeners. Also, we really want to look at your perspective, which will trigger, I think, some of the thoughts in us also, and maybe we are able to transmit them to others as well. So, Professor Gagandeep Kang, the floor is yours now. Yes.
Thank you very much, Dr. Katoch, for the very generous introduction. If you can tell me whether my slides are visible, I think I could get started. So can you see my slides?
Yeah, they are visible, but they're not full screen. They are visible, but not full screen.
Okay, it's full screen on my screen.
It's full screen. It's full screen. I can see it's full screen.
Okay. So thank you. I'm going to be talking about the current and future promise of SARS-CoV-2 vaccines. Very vast topic and lots to discuss in that area. But I hope to do this in less than the allotted time. So maybe we have some time for discussion after that. So I think the important thing to remember is that we actually have a lot of defenses. And today we are not where we were at this time last year. And while there are a lot of naysayers, particularly in the past couple of weeks about what is to come, I think we should be thinking about the situation, preparing for it, but also reasonably positive about what's likely to happen. So if we look at confirmed cases in India and the world, you can see that we had 2 peaks in India, and then we've had this very long tail from June onwards, where we've had quite few cases in the country. So sharp drop and then a tail, including the little bump that was large numbers of cases in Kerala. When we look at the situation where the rest of the world is concerned, you can see that there is a lot of increased cases and transmission that's happening in the Americas, in Russia and South Africa, and with the introduction of Omicron cases being reported from Australia as well. But in India, we have not seen a large number of cases at this time. Now, just as a reminder, the virus that we are dealing with, most of the vaccines that we have are based on the spike protein. And if we look at the spike protein, it's been important to think about making vaccines from a point of view of the structure of the spike protein and how it interacts with the ACE2 receptor. Very early on, from work that was done with the MERS coronavirus, it was recognized that the receptor binding domain of the spike protein was found in two positions, either in what's called a lying down position or in a standing up position. And in the lying down position, you have lower receptor binding and the possibility of immune evasion. And with the standing up position, you have higher receptor binding and greater immune recognition. So some of the vaccines, particularly the mRNA vaccine, which has been shown to work reasonably well against a range of different variants, has the spike stabilized in the standing up position. So I'd like you to hold that thought. Now, the other thing to remember is that when we look at vaccines for mucosal pathogens, we have to look at two aspects of immunity. One is systemic immunity and the other is mucosal immunity. When we get an infection, we get both systemic and mucosal immunity. However, when we get vaccines, all of the vaccines that we have at the moment are vaccines that are given parenterally. So therefore, we get largely systemic immunity. There are vaccines in development that are looking at the mucosal component, but those have not yet been approved or are available now. If we look at the immune responses that are generated by vaccines, here are examples, first of the mRNA vaccine and second of an adenovirus vectored vaccine. Essentially, both of these deliver the message about how to make the spike protein into the cell. This results in the cell expressing the spike protein on the surface and releasing some spike protein from the surface. And this results in the production of signaling molecules, cytokines and chemokines, and the stimulation of both T and B cells. Now, when you have an interaction of the T cell with the T cell receptor interacting with MHC class 2 molecules, you then have a signaling mechanism that results in the production of high levels of antibodies by B cells and their transformation to plasma cells. A proportion of the T cells and a proportion of the B cells also become memory cells, and these reside in various locations in the body, including the germinal centers of lymph nodes. Now, the other important thing to recognize is that there is not one class of antibodies that is produced. Most of the commercially available assays that everybody is going out and measuring their antibody responses to really measure only binding antibodies. And it's also very important to remember that among the binding antibody assays, these assays were established to measure binding antibodies following infection. And following vaccination, you may not quite have the same kind of response as you do following infection. So there are new assays that have been developed even for binding antibodies. These are not yet widely available, and they are actually better assays for measuring responses to vaccines. However, when we look at vaccination, we want to look at both a binding antibody response as a measure of immunogenicity, but also at neutralizing antibody responses as a measure of whether there are functional antibodies or not. And there are different types of neutralizing antibodies to SARS-CoV-2. When we do studies that there are 4 classes, depending on where the neutralizing antibodies respond, as shown in the figure at the bottom. And with different kinds of variants, what we tend to see is a difference in which classes of antibody responses are affected by the variant causing an infection. So for Omicron, for example, based on genomic data, we are expecting to see a big drop in neutralization activity. And that has been seen now with sera from both Pfizer and the AstraZeneca vaccinated individuals, that there is a drop of as much as 40 times in the neutralizing activity. But of course, neutralizing activity on its own, measured in vitro, does not necessarily indicate that those vaccines will be completely ineffective. Now, how is the neutralization assay set up? Well, a lot of places are doing a pseudovirus neutralization assay, which is not exactly the same as a wild type neutralization assay. But essentially what we do is, so far we have been taking serum or plasma from individuals who have been vaccinated with a range of different vaccines. And we have added either the live virus or the pseudovirus, and then seen whether we can interrupt the infection of these live viruses or pseudoviruses into cells. If that happens, that means that the antibodies that are present are functional, they are neutralizing either the virus or the pseudovirus and preventing its entry into cells. This can have a range of different kinds of readouts. You can have fluorescent readouts, or you can do microneutralization tests that look at the presence or absence of cytopathic effects. Or the most complicated, of course, is to do plaque reduction neutralization tests that are not done in microtiter plates, but in larger plates. Those give you the most accurate readouts, but are rarely being done nowadays because of the speed at which we need results from these kinds of studies. Now, when we've looked at neutralizing antibodies across a range of vaccines, what we've seen is that there is no absolute cutoff, no way of saying that antibodies above this level are going to guarantee protection against any variant of the virus. But we do see that neutralizing antibodies, at least in the data that has been generated from the phase 3 trials, do correlate with the level of protection that is seen with various vaccines. In general, vaccines that are based on the mRNA platforms produce highest levels of both binding and neutralization antibodies. And in general, the inactivated vaccines produce the lowest levels of binding and neutralizing antibodies. Covaxin, possibly because it has an adjuvant that is different from the adjuvant present in other inactivated vaccines, gives a higher neutralizing antibody response based on the data that has been published for the studies done by Bharat Biotech. Now, if we look at the platforms that are available for vaccines, there are essentially 6 kinds of platforms. We don't yet have live attenuated vaccines, but we do have inactivated whole virus vaccines. We have virus-like particles, recombinant proteins, vectored vaccines, DNA vaccines, and RNA vaccines. In India, currently, we have inactivated vaccines, vectored vaccines, and DNA vaccines that have been approved by the regulator, and we have other platforms that are on their way. If we look at the global picture and look at where we are in terms of studies, we have preclinical phase 1 safety and dosage, phase 2 expanded safety and immunogenicity, phase 3 efficacy studies. And as you can see in the blue boxes at the bottom, there are still many different kinds of vaccine candidates that are in development or have been approved for use. 25 vaccines are in use in different parts of the world, and 10 vaccines have been abandoned after entering human studies. Now, last year in May, WHO had developed a target product profile for vaccines, and what they wanted was 50% efficacy, not more than 2 doses, preferably. A vaccine that was usable in adults and gave protection for at least 6 months. And if we look at the list of vaccines on the right-hand side, these are all vaccines with efficacy data. A range of different kinds of platforms, mRNA, DNA, adenovirus vectored, inactivated, conjugate vaccines that have been developed in combination with tetanus toxoid, as well as recombinant DNA or subunit vaccines. Now, if we look at vaccination in the world and in India, from our world in data, we know that 56% of the world population has received at least 1 dose of COVID-19 vaccines. 8.5 billion doses of vaccine have been given globally, and 36 million doses are given every day. However, only 7% of people in low-income countries have received 1 dose. If we look at the figures for India, 1.34 billion doses of vaccine have been given in India. We are somewhere between 5 and 7 million doses a day. We've given out, as of this morning, 821 million first doses and 525 million second doses of COVID-19 vaccines. And shown here by doses is the CoVIN app data, showing total 1 and 2 dose coverage by week since the vaccination program started. Now, if we look at cumulative doses that have been given around the world, India ranks second. Of course, it's important to remember that only 2 countries in the world need to give more than 1 billion doses, China and India. And both those countries have given out 1 billion doses. Now there are some doubts about China and its needs to give more doses, because the bulk of the Chinese vaccination has been with inactivated vaccines. If we look at vaccine doses in terms of coverage, then we are doing very well given the size of our population, but we still have some distance to go, since 15% of adults have not received even a single dose of vaccine as yet in India. Now, if we look at our place compared to the rest of the world, what's shown here is the share of people fully vaccinated, meaning 2 doses, or partly vaccinated against COVID-19. In India, we have about 59% of the full population, not just adults, that have received 1 dose of vaccine, compared to 56% of the world. But what is worrying is that we have a greater gap between first and second doses compared to the rest of the world. And this has, of course, been highlighted in media reports as well. If we look at the choice of vaccines available in India today, we have Covishield, Covaxin, and Sputnik. About 88% of what is used in has been used in India as Covishield, about 12%, little less than 12%. Is Covaxin, and Sputnik has been very limited use. I think it's important to emphasize that even though we are using Covishield in India, there are many high-income countries that have chosen not to use Covishield in their younger populations, under 40, varying age groups, because of the risk of thrombosis with thrombocytopenia syndrome. This has been reported at a rate of anywhere between 1 in 30,000 to 1 in 200,000 vaccinated individuals, younger individuals in countries that did use these vaccines. But in India, we have very, very few cases of thrombosis with thrombocytopenia syndrome that have been certified as such by our national AEFI committee. Now, future vaccines in India, this is a matter of great pride that every platform being done anywhere in the world is available in India. And we have many, many more vaccines to come. Zydus Cadila did a phase 3 trial of 28,000 participants. This vaccine has been approved and will be included in the program soon. And the next 3 vaccines that we have coming up are one from Biologically, Corbevax, Genova's mRNA vaccine, and Covovax, which is the Novovax vaccine being made by Serum Institute of India. This has actually been approved by regulators in Indonesia and the Philippines, and hopefully will be approved in India as well. The J&J vaccine is being made by BioE, but currently it's being made for export because we are still having discussions on issues of indemnity for the vaccines that are made by or for international manufacturers like Moderna, Pfizer, and J&J. Now, if we look at the impact of vaccines, I'd like you to look at the inverted pyramid first. For vaccines, we expect the greatest efficacy or effectiveness of vaccines against death, then against ICU admissions, then against hospitalizations, then against mild disease, and finally against infections. Now, for infections, it's very important to remember that for a mucosal pathogen, we should not expect to see full or even partial protection against infection. It was, in fact, a bit of a surprise with the mRNA vaccines when the early data from Israel showed a very high level of protection against infection. But of course, when those individuals who were vaccinated were followed up, after a while, they started to see infections in them. Now, there are different ways of looking at the impact of vaccines. And to me, the ones that are most important are the top ones, which is what happens to prevent death, what happens to prevent ICU admissions or severe disease or hospitalizations, and only after that, the rest of mild or moderate symptomatic infections. And the other important thing when we look at impact of vaccines is to look to see are vaccines working the same way against variants. Now, the data for Omicron is emerging, but just as an illustration, I have the data here for comparing Alpha and Delta. And this is data that came out from England, Scotland, Israel, and Canada. What you can see here is while there was a lot of noise made about how vaccines would not work against Delta, the drop was actually greater against symptomatic infection than against hospitalization. So the vaccines all worked really pretty well against the severe end of the spectrum with the variants that were studied. So I think it's important for us to remember that, and also remember that we are in the early days of a new variant. And again, to illustrate why we need to think about boosters and think about variants, I have data here from Israel. And the first thing to look at is the orange and blue graph, where you can see that until about May and half of June, mostly the virus that was prevalent in Israel was Alpha. And when Delta came in May, Delta really took over and became almost 100% of the variants that were seen. Now, when you started to look at what was happening in terms of infection rates, which is the histogram on top, what you see is that there was waning immunity, and the way Israel set up their analysis was to look at people in terms of when they were vaccinated and what happened to infection rates among them. What you see in the white bars is healthcare workers and people at risk who received vaccine first. And then you start to see people who received their vaccines at various points in January, February, March, etc. And if you look at infection rates per 1,000 population, what you see is that people who were vaccinated earlier, healthcare workers, people at risk, as well as populations that were vaccinated in January and February, saw much higher rates of infection than people who were vaccinated at later time points. And this was true across all age groups. They also looked to see whether you could, what was happening in terms of severe cases. And what they found was that when Delta came in, they started to see an increase in the number of active severe patients that they were seeing, in whether they looked then to see whether these were vaccinated or unvaccinated individuals, and they found that they were seeing severe infections in vaccinated individuals. So then they introduced the third dose, and once they introduced the third dose, they looked to see what was the impact of the third dose, and found that the third dose reduced infections and severe infections in people who had received booster vaccination. As you can see, decision making for Israel required a lot of analysis of real-time data that allowed them to analyze what was happening in terms of infections, in terms of hospitalizations, stratified by age. Now we come to what's front and center of everybody's attention where SARS-CoV-2 is concerned, what's going to happen with Omicron. And the reason why people started to worry about Omicron very early on was that this had more mutations and constellations of mutations than had been seen in any variant so far. So as soon as South Africa described how many mutations there were and where they were located, WHO was able to rapidly declare this a variant of concern. We are now 3 weeks into this, and this variant can be found all over the world. But I think one thing that is very important for us to remember is that even though we have had variants that have spread widely in the world before, not all countries have followed identical patterns. So what you see here is in shades of green are Delta variants of various kinds. What you should see in the dark pink is the Alpha variant. What you see in purple is Omicron. And if you look at the United Kingdom, you have the D614G in orange, you have the Alpha variant in the dark pink, followed by Delta coming in around June and taking over pretty much by July. So this was the pattern in the United Kingdom. In India, even though we had an introduction of Alpha in some cases in December, but then mostly in January, the Alpha was replaced very, very, very quickly by the Delta variant. If we look at what happened in South Africa, you saw the Beta variant in the bright red, very little of the Alpha variant, which was the most transmissible variant that had been seen up to that point, and a rapid replacement of the beta and alpha by the Delta variant, and then its replacement by Omicron. So not all geographies have behaved the same way, even with very transmissible variants. In South Africa, we started to see a very rapid rise and were able to track that because of S gene target failures that showed us a very rapid increase over all provinces of South Africa. We have data that seems to be showing us that where Omicron is concerned, we are seeing cases that are less severe. But I would still wait till we have tens of thousands of cases before I'm absolutely convinced that we are going to see a pattern of severity that is going to be different from the variants we have seen in the past without accounting for vaccination. If we look at Omicron, it's certainly grown faster, and there does seem to be a divergence between the case detection and the hospitalizations. I hope that this will continue to be maintained, and this may be an intrinsic feature of the virus, or it may be that this virus is currently being seen in populations that are highly vaccinated, and therefore there is an attenuation of the severity of disease. Data continue to emerge about that, but certainly in terms of transmission, we know that this is becoming dominant in places where it has been seeded, and we should expect to continue to see this pattern in many more locations around the world. In India, I'm sorry, this is outdated now, but the S gene target failure in UK has increased very rapidly, and we are we are seeing super spreading events in multiple locations. What do these mutations mean for immune response? We've already seen that there is a reduction in the neutralizing antibody responses. And this was actually predicted by the mutations that had been seen and were expected to have a strong antigenic effect. There have been no other variants that have had 4 mutations that would have predicted a reduction in the neutralizing antibody response. So, but one thing I think it's important for us to remember is that with all of the variants and all of the studies that have been done so far, most of them have been done in relatively small numbers of samples from people that have received 2 doses of a range of different vaccines. But it's still unlikely that we will lose all of our antibody-mediated protection or all of our protection. When we think about vaccine effectiveness against new variants, there are 3 things really to consider. One is whether antibodies recognize and neutralize these new variants. These data are generated in the lab, and therefore much of what we've seen in the last week is this kind of data. The second is, are there cross-reactive cellular immune responses to this variant? And the early data that has come out from the Assay lab is reassuring in telling us that about 90% of our cellular immune responses seem to be maintained. And third, and most importantly, the data that we are really awaiting is the real-world impact of the Omicron variant on clinical protection that is provided by vaccines. The UK has published early data Friday and Saturday last week, indicating that if you've had 3 doses of the mRNA vaccine, you have a reasonable level of protection against severe disease. And if you've had 2 doses of the AstraZeneca vaccine followed by 1 dose of the mRNA vaccine, you also have reasonable protection against severe disease. But there is still data that we need to get from the Omicron variant to show us exactly how it compares with the variants that we have greater familiarity with. I think it's still important for us to remember that these data take a while to generate, specifically against variants, because large numbers need to be studied. And this so far has been done only in high-income countries. Despite the fact that China has given out so many doses of vaccine, India has given out so many doses of vaccine, we actually do not have data on the performance of the other vaccines that are broadly in use in our countries. The Sinovac data, for example, is data that came out from South America and not China. You know, if we begin to look at the impact of variants of concern on vaccine response, where we should start with is looking at the blue figures, which is protection against infection. And protection against infection is likely to be the least maintained. If we look at neutralizing antibody responses, what we should expect to see is a rise and then a decline, which will go up if we give booster doses. And neutralizing antibodies appear to be important to protect us against symptomatic infection. However, the fact that our cellular responses are maintained indicate that we should most likely expect to have some level of protection against severe illness, hospitalizations, and deaths, with most of the variants that we have seen so far or will see in the future. I'd like to just come to nearly the end to say I think it's really important for us to trust our immune systems and understand that infection and vaccination both have a role to play. We know with the early data from Omicron that what people are currently calling hybrid protection, which is infection plus vaccination, gives a good level of protection, and that promises good things for India because about 50% of our population has been infected and then has vaccination on top of that. Now, T-cell responses, the SETI lab indicates very limited losses, and I think it's important for us to remember that while virus neutralization is desirable and we want as much of it as we can get, it's not necessarily essential for protection against severe disease. So there are many issues with vaccines. Which vaccine is best? What are dosing intervals? Do we need boosters? Should children be vaccinated? What are contraindications? And I'll just take you very quickly through these. Which vaccine is best? Bottom line is that the emerging evidence from other parts of the world seems to indicate that we should consider a mix and match strategy when it comes to boosters. The problem is that the better vaccines are so far not available in India, so it has not been possible for us to study them. If we look at dosing intervals, I think a lot of controversy about this, but a longer gap in general is better, as long as we don't need that marginal increase in protection urgently. And this is the reason why the UK shortened its dose when the Delta variant came around. Are boosters necessary? Absolutely, immediately for those who are immunocompromised and possibly likely for the over 60 elderly. For Omicron, what we know is 3 doses of mRNA are definitely better than 2. And 2 doses of AstraZeneca followed by mRNA give 71% protection. In India, we currently have 3 vaccines with more to come. We have no data on waning immunity. The neutralizing antibody data from other parts of the world seems to indicate that Covishield may need boosting. There's no data on Covaxin, but in terms for Omicron as yet, WHO has recommended boosting people with inactivated vaccines. We don't know if that's required for Covaxin. As I mentioned, it does have a different adjuvant. It's possible that at some point in the future, we will have the data to tell us who needs vaccines, and maybe we will have better vaccines as well. Should children be vaccinated? Immunocompromised or with comorbidities? Yes, absolutely now. But others, I'm not sure that we actually have data telling us that we need to vaccinate children immediately at this time. And this is obviously very controversial. But I think we need better data, and we need to remember that we need to set up our children to be protected for as long as possible, which means that we have to consider original antigenic sin and the fact that we might be shaping the immune response of our children for some time to come. The other important thing to remember, of course, is that about 60% of our children are already infected. Contraindications, there are several. And again, it really bothers me that we don't have very good safety monitoring in India. These are new vaccines, and to have confidence in the vaccination program, safety is a critical concern. However, when we look at what our National AEFI committee does, is it reports very infrequently, its reports are delayed, and it becomes very difficult to find any cases for which causality is assigned. Most of the cases that they seem to have reported to them seem to be anxiety-related cases rather than actual other findings that have been described in other parts of the world. So this is an area for us to concentrate on. To summarize, vaccines work. Current vaccines are likely to work, but for some variants, we may need to vary our vaccination strategy. Data is key for decision making, and there's very limited availability of relevant data in India. Safety monitoring in our country is limited and delayed. Platform technologies where new vaccines can be rapidly made should be established in India at scale. We have one of each. We should be thinking about having many more players in the mRNA and DNA and viral vectored vaccine space. And regulatory processes need to keep pace with innovation. And finally, I think pan-coronavirus vaccines should be a focus for research and development, because these will allow us to get away from needing to react separately to each variant. So with that, thank you very much for your attention.
Thank you, Professor Kang, for such a condensed lecture, actually, that you have covered almost everything. See, we have, I think, 3-4 minutes. We'll have the quick questions. First point which I would like to flag to you is about the safety issue, the monitoring part, which concerned the people in the beginning also. But on the scale, the vaccines have been used in India. Maybe, the very few side effects have been reported. Otherwise, if they must be occurring, but they may be at a low level. But how to strengthen the system? That's something which is a very important from a person like you who is a concern on the issue. Yes.
So I think there are two approaches. One is, we have to make people understand that when you introduce a new vaccine, you're really looking for side effects that you cannot find in a phase 3 trial, because phase 3 trials are too small to detect rare side effects. But unfortunately, because our routine vaccination program, most of the side effects have not been vaccine product related. They have been vaccination related. Injection abscesses, mixing the wrong adjuvant, etc. The mindset is to not report AEFIs. So educating people that are involved in the program that we are looking for vaccine product related effects is critically important, and that has not been done. I think there are
I had a
efforts
question.
always to strengthen it, but they haven't had impact yet.
Could I ask a
Thank you,
question,
Dr.
Dr.
Kang. Dr.
Katoch?
Ghosh? Yes, Dr. Ghosh. Yes.
See, from your lecture, Dr. Kang, I got the impression that booster doses are very essential. But I see a reluctance on the part of our government. What could be the reason? I don't know. It's a fair question to ask, but
I think it's about a lack. So I don't know why the government might be reluctant, but I know that from thinking about how to make decisions about boosters, you need data. And the primary data you need is vaccination history of people who test positive and have severe disease. And we don't have that. We have cases, so you know who's tested positive, but you don't know whether they have symptoms or they don't have symptoms, how bad their disease is, nothing. And then you have data on deaths. So for deaths, there is some vaccination information, but deaths are rare. And what you really want to study is effect against moderate and severe disease, and we don't have data. That's where you will see waning. And we're not seeing it because we are measuring a rare endpoint.
Thank you.
thank
Thank you,
you.
Doctor. Dr. Habib, you raised your hand earlier.
Yes, so I just had a quick question, Dr. Kang. The platform of RNA vaccines has been sort of said that, it's so useful because you can modify it so quickly, and you can, if there's a variant of concern or something, you can modify it very quickly and bring it, into use. So what is being done with that platform in India regarding Omicron and the mutants associated with it? Is there a move to modify it and use it?
So, well, I have as much information as you do. Saman comes from the papers, and it's Adar Poonawalla saying they are planning to modify the vectored vaccine platform. That is actually being done in Oxford. They have already made an Omicron viral vectored vaccine, and it's also been done by Moderna and Pfizer. So wherever it's sequence-based, message-based vaccines, we can say, it's easy to modify those vaccines, and it has already been done. In
Thank you,
India,
sir.
the only mRNA vaccine player is Genova, and their technology is transferred to them from Seattle, HDT. So I don't know if they have plans to modify it, but I'd be surprised if they did not.
Pastor Mehra? Yes.
Yes, I must thank Dr. Gagandeep Kang for a brilliant lecture. I really enjoyed it. Gagandeep, I have one question. In terms of the children, the vaccination in children, the world data shows that the severe illness is rather uncommon in the children. This is also from the immunological point of view, yet there are benefits of vaccinating children, and that everybody knows in terms of protecting them, decreasing spread to the families and communities, and ensure the educational continuity over there. So, given the Omicron existence now, would you really recommend that we should not wait any more longer, but go ahead and give the vaccination to the children? And I will come back to my second one after you answer this.
Okay, so if you have 100 doses of vaccine and you need 90 of them for your routine program and you have 10 left over, if you have elderly people who need a booster dose, I would go to the elderly people with the booster dose first before I would go to vaccinating children. There are modeling studies that have been done by a number of groups that say that for prevention of severe disease, it's much better if you have a limited number of doses to vaccinate the elderly and those with comorbidities rather than vaccinating children. Now, that is an analysis that is done in the context of a limited number of doses. Does that mean that you should not vaccinate children? If you have unlimited doses, absolutely. These are safe vaccines. And if we do it in an age descending strategy, I think it's fine to vaccinate children, because even if severe disease is very rare, it still exists. It's not zero.
And my second point was in terms of the booster dose, or what we call in India additional dose or a third dose. And the data you showed from Israel, and the data is also available from UK now, that if you mix mRNA-based vaccine with, say, AstraZeneca, that the boost is much higher.
Yes.
After the third dose. We do not yet have the mRNA vaccine in India, although we are looking forward to Genova and Sanjay Singh, with bated breath for a long time. Hopefully that will come. But if it doesn't come, what would you recommend with the existing structure that we have, only with Covishield and Covaxin? The people who have taken 2 doses of Covishield, should they take the Covaxin or the other way around?
So we are currently doing a study. My colleague Winsley Rose is leading the study. We anticipate having our results by the end of January. In fact, our Covishield arm is finished, but the Covaxin arm has been hard to recruit for. We hope to finish recruitment by the end of
Parthasarathy and Dr. Brahmachari, one question each. I think we are. Thank you, sir.
Thank you. My question asked by Professor Mehra. I got the answer.
Thank you, sir. Thank you, Dr. Brahmachari, sir. Your last question from you.
No. The BM, she was answering that in terms of the Covishield and Covaxin. She said.
Okay. Madam, continue, Dr. Kang. Yes.
No, I'm just saying there will be data by end of January, but we may need to make decisions before that, or, if it's in the interest of people to unblind before that, maybe we could think about it. Depends on the urgency over the next 3 or 4 weeks, and only if our DSMB and the government want the data. You're on mute, Dr. Brahmachari.
Gagandeep, since you mentioned that our Pseudo survey shows a very significant percentage of the children are also infected, can we link the antibody check before we give a booster vaccination? Or well, I got my antibody profile, my wife's antibody profile. She has 10 times or 20 times more than me. So why to vaccinate her? Give a booster. Why not I get the booster? So I'm just saying it will not be a more prudent decision, because over pumping the immune system is not also a very good idea, especially for children. How do you think about it? And so far, mRNA vaccination is concerned, already for women with young women has severe problems with respect to their menstruation cycle and all. Already there are studies. So is it a good idea to push messenger RNA vaccine for Indian young women?
So there's children, there's young women, and there's elderly, and there's different kinds of vaccines. So for children, in general, it appears that even adolescents might benefit from vaccination. The question is 1 dose or 2 doses and which vaccine to give. I don't think there is any urgent need to make decisions on that, and I would prefer the least number of doses of the best vaccine. I'm not sure that Covishield and Covaxin are the best vaccines for our children. So I'm willing to wait on that. Now, should you make antibody-based decisions? Well, if you had a cutoff that showed you protection, maybe it would be useful, but we don't currently have that. It gives a certain level of reassurance to know that you've got high antibodies, because the likelihood is that you will be protected against that. I'm very clear that it's the immunocompromised, the elderly, and those with comorbidities that must be prioritized for completing primary immunization and getting booster doses. After that, we think about younger populations.
Thank you, Professor Kang, for a So nice of you for I think answering all the same time. Fun targets. Very much, Kang. Thank you. Well, we go to the next speakers. Dr. Saman Habib. I think again each of us at the moment from the CDRI, but I mean that's a for her, for many of us, you know, are actually the images from the CDRI. I think her in the malarial biology actually they are superb. You know, her work on the mitochondria, her work on Epicoplast, her work on the genetics, her work on the protection. I think she has in these 3 decades of career, you know, that she has touched everything with very impactful science. And can it reduce the malarial parasite? Dr. Saman Habib, please.
Thank you, sir. I'll just share my slides quickly. Can you see them?
Yes, we can.
Are they visible? Yes. So I'll just...
Yeah, we see them. Yeah.
All right. Thank you. So thank you very much for introducing me like this. And I hope I'll finish my presentation in time so that there's some time for a bit of discussion. As we know, malaria continues to be a disease of concern because there is rising resistance to frontline anti-malarials across the world. And also because finding a vaccine for malaria has been such a challenge, and we still don't have a very efficacious vaccine for the disease. Apart from the fact that our interest in finding new drugs and new vaccines, research interest in the parasite has also been sustained because of the unique biology of this parasite. So if you look at the way malaria infects, you know, it is able to have this elaborate life cycle in the human and part of its life cycle in the mosquito. And both these is when it adapts to very different environments in the human and the mosquito. So it's almost like the parasite living in 2 different planets, and it's able to make this change. It not only changes morphologically with these multiple stages, it's got this extremely complicated gene regulation system with different kinds of genes being activated at different time points in its life cycle. There is complex regulation in these steps, and then there is also this business of immune evasion, because of which it's so difficult to get a vaccine for malaria. Another level of complexity in the parasite is not at this stage of the life cycle, but also in the cellular level, and that is in the kinds of organelles it has. And if one looks at the way this was discovered, it was interesting because in the early 90s, and this is around the time that I was doing my PhD and finishing my work in around 96, in 1991, there was a report that said that does the malaria parasite have 3 genomes, which essentially meant that people were asking the question that apart from the nuclear genome and the mitochondrial genome, is there a 3rd genome seen in the malaria parasite? And this turned out that when this genome was sequenced, so actually the genome was identified first before the organelle to which the genome belonged was identified. And when this genome was sequenced, it turned out that the sequence of this DNA, the 35 kb circular DNA, was extremely similar to plant plastid genomes, which raised the question in 1997 by McFadden and Waller that whether the malaria parasite was a plant. And this was around the time I was finishing my PhD and joined CDRI and started looking at this organelle and its functions. And this was a very exciting field for us at that time. But if you look at the way of the mitochondrial genome, so this is the Epicoplast genome to my left, the 35 kb circular genome. But the other genome apart from the organellar genome is a mitochondrial genome, which is the smallest known mitochondrial genome in nature. It's only 6 kb long. It codes for only 3 genes, which is cytochrome oxidase 1 and 3, and cytochrome B. And it encodes for a series of fragmented ribosomal RNAs, which assemble to give rise to a very, very small and distinct ribosome, which I will talk about eventually. Now, if you look at the way these organelles are organized, the epicoplast has 4 membranes surrounding it, not 2 membranes like we know for plant chloroplasts, but 4 membranes surrounding it. And the mitochondria has the usual 2 membranes surrounding it. And most of the physiological pathways, metabolic pathways that take place, and I'll come to them eventually, in the epicoplast and the mitochondria are carried out not by genes encoded by these reduced genomes, but by proteins that are made in this nuclear encoded, made in the cytosol, and then are transported or imported into the organelles. And these require specific signals for import, and these have been characterized. So what is this, and what sort of evolutionary insight does the presence of this epicoplast give us in terms of understanding how parasites have evolved? So if this four-membrane organelle made people understand that it's a result of a secondary endosymbiotic event. So if you try and understand how this organelle came into being, we now know that ancestral cyanobacteria must have been engulfed by a first phagocytic eukaryote, as a result of which you had this dead alga-like organism. And if this dead alga-like organism would be eaten up by a second phagocytic eukaryote, you would have a generation of a nucleomorph. This nucleus would eventually disappear and assimilate into the host nucleus. And one would have this sort of organelle, which would be surrounded by four membranes. The first two membranes being the original cyanobacterial membranes. This membrane being the membrane that came from the phagosome that had engulfed this here, and the outer membrane being of the parent plasma membrane. So if this happened, what we now saw in terms of the malaria parasite was actually an organelle that had lost photosynthesis. So these thylakoids that you see here, they do not exist anymore. So the photosynthetic machinery has been completely lost in this organelle. This was a hypothesis till the time in 2008, as late as that, off the coast of Australia, endosymbiotic with corals, was discovered another set of organisms called chromerids. And these chromerids were very similar to what we know as Epicomplexa, to which the Plasmodium belongs, except that in their plastid, they had retained photosynthesis. So now we had a missing link between this and this, and we knew that there is this class of organisms in which the plastid-like structure exists with four membranes surrounding it and retains photosynthesis, whereas in the subsequent epicomplexan class, photosynthesis has been lost, and we have what is now called the epicoplast. Now, if you look at epicoplast morphology, it changes drastically as the whole. As in the RBCs, you see a very small epicoplast early on, the single organelle in the parasite, just like mitochondrion, which is a single organelle in the malaria parasite. So each cell of the malaria parasite has only a single mitochondrion and a single epicoplast. And as they start dividing, you see a very long elongated mitochondrion and a long elongated epicoplast. And as each merozoid is formed, each daughter cell receives a single organelle each. So if you look at the blood stages, this is an image from our lab. You can see this red, you have an elongated mitochondrion, two parasites infecting a single red blood cell. This is in red is the mitochondria, in green is the single epicoplast in each of these parasites. In the liver stages in the human, they adopt really complex structures. A single epicoplast seen here in green in an infected hepatocyte that is shown here adopts this extremely elongated and complex network, web-like network, and as does the mitochondrion. And they became extremely large single organelles that have this very interesting mesh-like structure. And then as each daughter cell is formed, these go as one organelle each in a daughter cell. So if one looks at how epicomplexa have evolved in terms of understanding the plastid and the role photosynthesis has played in this evolution, one can imagine that they come from a cyanobacterium, which is 2.8 billion years ago. Then they became primary endosymbionts in a red alga. Red alga was engulfed by a eukaryote around 800 million years ago. This gave rise to a chromangiovate like in Chromarabilia that we see here is photosynthetic. Further on, they adapted to marine organisms, then to terrestrial organisms where they became parasitic, and finally to humans where parasitism was established. And then the vector came into the picture where you have transmission which takes place through the vector. So Toxoplasma, Plasmodium, both epicomplexan parasites and others have an epicoplast, but Cryptosporidium, which is also an epicomplexan parasite and is very important for diarrheal diseases. In India, around 7% of diarrhea in children is caused by Cryptosporidium, lacks the epicoplast. So you have a loss as well. So in some organisms, in some epicoplexan parasite, it's been retained, and in one like Cryptosporidium, it's been lost. So there's been this change from free-living heterotrophs to obligate intracellular parasites, and you see this transition happening across evolution. So when we started working, we asked some questions on what this epicoplast was doing, what this genome function was it actually a translationally active organelle? And our lab was the first to show that the epicoplast is translationally active, that the genes encoded, the limited number of genes encoded in its genome are actually translated, and we were able to localize these and also show that translation inhibitors can actually inhibit active translation and cause parasite death. So the question was then asked that because it's got a prokaryotic origin, and therefore got prokaryotic systems or prokaryotic mechanisms of translation, can antibiotics target Epicoplast translation and cause parasite death? So chiromycin, that targets elongation factor 2U of translation, exhibits antimalarial activity in culture. And inhibitors of prokaryotic translations such as chloramphenicol or thiostrepton that we showed ourselves and clindamycin are all believed to target Epicoplast translation and cause parasite death. But the question that needed to be asked was that there were two organelles of prokaryotic origin: mitochondria that came from alpha proteobacteria and epicoplasts that came from cyanobacteria. And therefore, the question was that if you're using antibiotics and claiming that these kill the parasite, what were they targeting? Were they targeting mitochondrial translation or were they targeting epicoplast translation? So we devised a series of experiments in which we expressed all these translation proteins. I'm not describing those in detail, but essentially what we tried to understand was where were these antibiotics acting. And it turned out, with an example from fusidic acid here, that this steroid antibiotic had a much greater inhibitory effect on the epicoplast translation in EFG than it had on mitochondrial EFG. And this was shown, this was because there was an insertion in the mitochondrial EFG that altered the way it bound to the antibiotic. Now, currently, if you look at what's of use of antimalarials in use, doxycycline, which binds the small ribosomal 30th subunit of the epicoplast ribosome, is in clinical use today as an antimalarial prophylactic. So you have an epicoplast targeting antibiotic in use, and clindamycin, which inhibits transpeptidation, is also in clinical trial for malaria, with another drug called fosmetomycin, which targets an epicoplast pathway, and I will come to that eventually. So you actually have inhibitors of epicoplast translation, epicoplast metabolism, which are in trial and also in use. Now, our lab had, this is a very cluttered slide, and I'm not going to describe this, but what I'm trying to say in this is that we, in a large consortium of labs under an EU project, worked on understanding how translation mechanisms work in both the epicoplast and the mitochondria. And of course, the idea was that can we actually inhibit these using different drugs? So Dr. Dominic Soldati's lab and also Dr. Amit Sharma's lab in India worked on this very interesting fact that in the mitochondria of the malaria parasite, amino acid tRNA synthetases are actually not imported into the organelle. Charged tRNAs are charged in the cytosol, and then charged tRNAs enter the organelle, which is just not seen in any other system. In any other system, you would have amino acid tRNAs also being imported into the organelle, tRNAs being imported or synthesized inside the organelle, and then charging of the amino acid tRNA taking place inside the organelle. But in malaria, this does not happen, and therefore there must be a system of import of charge and export of charged tRNAs, which is still not understood. There were several other differences. For instance, my lab found that there was no elongation factor TS in the mitochondrion, that the recycling of EFTU and TS took place at a slow pace, but did not require a GTP, a factor that would help recycle GTP and GDP. Similarly, the way the termination took place was different. The structure of the ribosomes was different in both these, and there were many such differences. So if one comes to the structure of ribosomes for now, we started looking at how these ribosomes were organized and what was their structure like, because it was clear that most of the ribosomal proteins would come from the nucleus. There were some proteins that were made by the epicoplast genome. And here I just show you a Venn diagram, wherein you see a comparison with P. falciparum and Toxoplasma gondii, which are both epicomplex and parasites. You see a comparison with diatom, a green alga, and a red alga. And as you can see here, a number of small and large subunits, which are expected from these to be there as part of ribosomes, are missing.
In the slides.
You didn't see a change?
No, first slide, we're still stuck in the first slide, I think. Second slide, the plastid and plasmodium.
Oh, should I come out of this thing and reshare? I'll just come back. I'll stop sharing and reshare maybe. Oh, I have moved quite ahead actually. Let me just stop sharing if that's okay. Just Can you see the slides now?
Yes.
The first slide.
Yes.
And then can you see the change?
Yes, I can see the change now. Yeah.
Okay, so I think I will not go to full screen. I think what's
Yeah.
happening is that the full screen is causing the trouble.
Okay.
Okay,
I can
so
see the one.
can you see this slide with this multiple
Yes,
engulfments being shown?
Yes, we can see that.
Okay, so this is where I was trying to say that there is this, I'm sorry, I wish somebody had told me earlier, I would have changed, that there is this process of secondary embiosis where secondary endosymbiosis.
And why not you make it a full screen?
If I make full screen, so what's happening is that it's not transitioning to the next.
Okay.
So I'll just show it like this, if it's all right.
Yes.
I'll try doing full screen again and see if it works. I've done full screen. Can you see it?
No.
Yeah, we
Not
see the
yet.
slide, but
I'm changing now. Can you see the change?
start.
No, we can't see any change. I think it's better that the way you were showing it before, because that.
Sure.
Yeah,
So so I'll just do that again then. Sorry.
Even without your slides, we were imagining everything so well. There was no difficulty.
No, that's not it.
No, you're explaining very, very well. Actually, there's no problem actually. Yes.
So this is where I just said that there was this secondary endosymbiosis event. And then I said that there was this transition from a cyanobacterium via chromalveolate to, you move on from interstitial to immunoorganisms.
Yeah, we can see that, yes.
And then I said that there was this thing of translation. You wanted to understand how translation takes place and, these multiple effects of antibiotics and how one had to understand whether it was Epicoplast or mitochondrion, and that there are these antibiotics in use for the malaria parasite because they target Epicoplast and mitochondria, Epicoplast translation mechanisms. And then I was trying to show here, this is the slide that I was at actually, that there is there are these differences that you see with known, which what we believe to be conserved translation mechanisms, and actually you don't see this conservation in the malaria parasite. They differ from prokaryotic, not only from prokaryotic translation mechanisms, but also from known whatever has been worked out in mammalian mitochondria in terms of mitochondrial translation. So there are big differences that you see in these organelle translation mechanisms, and that's what I was highlighting. And then I was here and trying to say that if you do this Venn diagram comparison of the way ribosomes are organized in the malaria parasite, and you here just compare the ribosomes in the epicoplast, you find that many subunits that are expected to be there in the ribosomes are missing in the malaria parasite and in both in Toxoplasma and in Plasmodium. So we did an analysis of the ribosomal composition, and if you just compare with E. coli, you find that in the mitochondria, in the epicoplast, for instance, this is the E. coli, this is a spinach plastid. You have a large number of ribosomal proteins here. You have 24 and 33 large and 24 small ribosomal proteins. And you have these many ribosomal RNAs. But if you look at the epicoplast, you have a much reduced number. You have only 16 versus 24, 24 versus 33, and you don't have the 5S and the 4.5S ribosomal RNA. And if you do modeling of this, you find it's organized somewhat like this. And in the mitochondrial ribosome, it's further reduced. The only ribosomal RNA you have is highly fragmented ribosomal RNA encoded by the mitochondrial genome itself, which tells you that this is a very, very reduced ribosomal assembly that you see here. In comparison to other mitoribosomes, this is really, really small. So this gives you a very interesting question on how this biogenesis of ribosomes take place and what are the functional implications of these reduced structures of organelle ribosomes, and how would these actually influence both interaction with antibiotics, how would these have altered interactions with translation factors, perhaps therefore they would influence the rate and regulation of translation, and then importantly, how do these ribosomes assemble? What are the assembly factors that are required for their biogenesis and for their maturity in parasite organelles? So the next question that I will address today is to understand how ribosome biogenesis takes place, because this large RNA protein complex, which has to assemble in a very sequential manner with multiple proteins and biogenesis factors interacting with it at multiple time points. Now, if one looks at, I will just here very briefly address only the small subunit of the ribosome in both the epicoplast and the mitochondria. So you have something called the Nomura Assembly Map, which was made by Nomura's group to understand how bacterial ribosomal small subunits assemble. So this is a 16S ribosomal RNA of the small subunit. And this is the primary assembly, the secondary assembly, and the tertiary assembly factors. And these are the small subunit proteins. The ones that are marked in pink and green just tell you that the ones in pink are there in the mitochondria, the ones in green are there in the epicoplast, and the ones that have both are present both in the mitochondria and the epicoplast. But what this tells you with this comparison here in Plasmodium is that some of them are present only in the epicoplast and absent in the mitochondria. Some are present only in the mitochondria and absent in the epicoplast. Many of them are absent in both. So there is a lot of differences from known systems. So we try to then understand how assembly takes place, and we sort of shortlisted a series of assembly factors and found that there were 7 GTPases, 14 chaperones and RNA maturation factors, and some dead box helicases that were putative organelle proteins and would be required for ribosome assembly to take place. Now, I will today just describe one of them to tell you the kind of multiple functions that these proteins can do. So if you look at this very interesting factor called PFKSGA, and you do a model, and this is a factor that has dimethyl transferase activity, and it adds it methylates 2 adenosine residues on a hairpin loop of the 16S ribosomal RNA in the small subunit. You do a comparison of this with other known KSGA-like proteins, and you find that the Plasmodium protein has an additional alpha-2 helix. Just keep this in mind when we come talk about it later. So we expressed it and raised antibodies and tried to localize it, and we found that it goes primarily to the mitochondrion, but it's also there. Some of it is also there in the cytosol, but it's basically a mitochondrial targeted protein where we showed it interacts specifically with the small subunit of the ribosomal RNA, and we could pull down this specifically and try and understand how much of it was binding where. And we found, in addition to binding the small subunit of the ribosomal RNA in the mitochondrion, we also found that it also bound to mitochondrial DNA by chip assays. And this was interesting because the functional role at the level of mitochondrial genome was not understood, and we just couldn't understand what it was doing there. So we did a series of analysis, and it turns out that this factor, this KSGA factor, has some similarity with yeast MTF1, which is the major transcription factor for the yeast RNA polymerase 41 in the mitochondrion. And it has some identity, some similarity with TFB2M of the human mitochondrial RNA polymerase. So this brought us to this whole question of how transcription will be regulated in the mitochondrial genome. I mentioned earlier that the mitochondrial genome is only 6 kb in length. We know from some experiments done in other labs that it's probably a monocistronic transcription system, that you have a single mRNA being made and then multiple proteins being formed after splicing. What was the start point of this transcription? What was the polymerase that mediated this transcription? What was the transcription factor that regulates this was completely unknown. So we analyzed first the RNA polymerase. We expressed the RNA polymerase partly. We raised antibodies and localized it. And as you can see over here, this extremely beautiful elongated mitochondrion and complete localization in the RBCs of the parasite. And then we did pull down assays with KSGA, and we found that yes, indeed, there was very specific RNA polymerase KSGA1 interaction, and that these two interacted with each other, both in the context of the DNA and without. So now we didn't have a functional RNA polymerase because it was a very large protein and we could express only a small domain. So we use a heterologous system, which is from yeast, and we got this from Pragna Patel's lab in Rutgers, wherein we use the yeast polymerase, the yeast mitochondrial polymerase with the yeast mitochondrial factor, but then also heterologous KSGA1 to try and understand if KSGA1 from the parasite could replace the yeast factor. And yes, in vitro transcription assays, this is with the yeast factor, the yeast polymerase. There is in vitro transcription activated only in the presence of this transcription factor. And KSGA1 can replace MTF1 completely, and you get very specific in vitro transcription. We did a SAC-CES structure and analysis, and we found that this alpha-2 alpha helix that we had shown earlier to be a distinctive helix is very important for the RNA polymerase KSGA1 interaction to happen. So this told us that the C-terminal domain, which has the alpha-2 helix, interacts with the N-terminal domain of the RNA polymerase, and that's how you get this very specific RNA polymerase transcription factor interaction in the parasite mitochondria. This also gave us a tool to understand the transcription start point of the mitochondrial genome, because now we could clone the entire mitochondrial genome in fragments and start doing in vitro transcription using the yeast polymerase and our factor. And as we used that and tested it, we found that you could get this transcription here, and we could really localize the transcription start site. So this was interesting. It helped us understand how mitochondrial transcription takes place, what is the protein required for it. And interestingly, this protein KSGA1 is not only a transcription factor, but also a ribosome assembly factor, and thus performs dual functions in the parasite mitochondria. And it differs, obviously, as I said, in major ways with the human KSGA1 as well as the KSGA1 known in other systems. That's as far as the housekeeping functions of these organelles is concerned. I mean, I described transcription, I did a bit of translation, and I tried to bring out some unique features of these organelles. But what do these organelles actually do? And why is the epicoplast so important for the parasite that even though it has such a reduced genome, it's been retained by the parasite? So if you look at the kinds of pathways that operate in the epicoplast, it carries out type 2 fatty acid biosynthesis, which is essential for parasite survival, not so much in the blood stages, but very majorly in the liver stages. It carries out heme synthesis, which is also interesting because there is excessive heme. It's in the RBC. There is so much heme in the parasite that the parasite active has to convert it into hemozoin to be able to survive. But this heme biosynthesis pathway does not function in the blood stages. It functions again in the liver stages and the mosquito stages. So one has to remember that. And the reason why the epicoplast is retained is because of the presence of the isoprenoid biosynthesis pathway that operates exclusively in the epicoplast and is the source for isoprenes, particularly IPP, because if you actually delete part of this pathway, the parasite dies. And IPP, if you supplement it with IPP, which is a product of the isoprenoid biosynthesis pathway, the parasite is able to survive even if the epicoplast is lost, telling us that isoprenoid biosynthesis is the major pathway because of which the epicoplast is essential for parasite survival. In addition, recent papers around 3 or 4 years ago saw a very interesting phenomenon. They showed that if you knock out part of the isoprenoid biosynthesis pathway, you would not get synthesis of the penultimate product, which is HMB-PP of this pathway. And if that happens, then the presence of increased carbon dioxide, aldehydes, and monoterpenes would not happen. So in essence, what they were saying was that the penultimate HMB-PP product of the epicoplast isoprenoid biosynthesis pathway causes an increase in carbon dioxide, aldehydes, and monoterpenes, which serve as attractants to mosquitoes. So if a person is infected with the malaria parasite and has this epicoplast, therefore, and this pathway is operational, then this results in a higher level of aldehydes and monoterpenes and carbon dioxide, which serve as chemoattractant for mosquitoes. And so if you have a large room full of people, of which 1 or 2 people are infected with the malaria parasite, probably mosquitoes or anopheles in that room will home in towards people who have the malaria parasite, and therefore this also has a very important role in transmission of disease. This gave a very interesting perspective to the reason why you would have an epicoplast in the parasite. So epicomplexans have retained the plastid because of their profound metabolic dependence on the endosymbiont. And the question that we ask is, can this become a liability for the parasite? So we tested this hypothesis because we noticed that in the epicoplast genome, there is one gene for the SUF pathway, SUFB. And the SUF pathway is a pathway for iron-sulfur complexation. Now, what is iron-sulfur complexation? Iron-sulfur complexation is the oldest post-translational modification known in nature. It probably was very important in the early reducing environment of the Earth. And as the environment of the Earth became more oxidizing, iron-sulfur complex post-translational modifications became more internal modifications on proteins. And largely, if you look at proteins now, they are part of very, very important proteins in DNA repair, in replication, in many, many metabolic pathways, etc. There are 2 major, and they function in redox and non-redox catalysis and in electron transfer reactions catalyzed by these proteins. So if you have this, you can have a 2Fe2 2-sulfur, 2 iron 2 sulfur cluster of this rhombic shape. You can have a 4 iron 4 sulfur cluster of this cuboid shape. And then these are coordinated on cysteine or histidine residues on the proteins that they assemble on. How are these iron sulfur clusters assembled on proteins that require them? So there are 3 major pathways. You have the ISC pathway, the NIF pathway, and the SUF pathway. The ISC system is found in bacteria, in prokaryotes, is found also in most eukaryotes. The NIF pathway is found primarily in nitrogen-fixing bacteria. But the SUF pathway, which is of interest today, is found in archaea, bacteria, protozoa, and plants. Notice here that it's absent in mammals and therefore absent in humans. So our argument and our hypothesis was that if it's absent in humans, can be there and been present in the malaria parasite, which is a protozoan, can we look at this pathway as a putative drug target for malaria? So we showed early on that the ISC and the SUF pathways in the parasite partition to the mitochondrion and the epicoplast respectively. The SUF pathway is specifically targeted to the epicoplast, and that's where the SUF pathway assembles iron sulfur clusters. It was shown around the same time that if you knock out one of the genes of the SUF pathway, then the parasite is not able to survive in the blood stages. So we characterized what this pathway was like, what were its constituents, and how it was working. So in general, you would require a sulfur mobilization from cysteine. You would also require an iron source. Then you would require a scaffold protein to assemble the iron sulfur cluster, and subsequently for the iron sulfur cluster to be transferred onto a target apoprotein or to many apoproteins, which might require transfer proteins. So we expressed what we thought were components of this pathway. We characterized their functions, and we understood that there was a cysteine desulfurase, which was SUF-S, which was encoded by the nuclear genome and was imported into the parasite. Similarly, there was SUF-E, which was nuclear encoded and imported into the epicoplast. And this was required for the mobilization of sulfur from cysteine with the conversion of cysteine to alanine. And SUF-E greatly enhanced SUF-S mobilization of sulfur. This was the structural model, and SUF-S requires a PLP, a pyridoxal phosphate as a cofactor. And if you use a drug such as dicycloserine to form a PLP cycloserine adduct, then you can actually inhibit this activity and cause parasite death. So we showed for the first time in principle that if you inhibit, so you can inhibit SUF-S activity, and if you inhibit SUF-S activity in the epicoplast and you inhibit iron sulfur synthesis, then the parasite is not able to survive. Then we characterized what the scaffold protein was like, and we understood that it had a SUF-B-CD stoichiometry in the ratio 1:2:1. So you had two SUF-C molecules, which were proteins, which were ATPases, and there was an interaction of SUF-D and SUF-C. This is the model that we created. And there was a specific binding site between SUF-C and SUF-D, and you could actually look at this binding pocket in terms of inhibitors as well. And only when you had the CD interaction would there be an activation of the ATPase activity of SUF-C. So to cut a long story short, we were essentially able to reconstruct the entire pathway. We were able to show how sulfur is mobilized. We still do not understand what the source of iron is precisely. We understood what the scaffold was like, what were the functions of each of these. We also understood which proteins transfer this 4Fe4S cluster onto the target apoproteins. And these are the many target apoproteins, some of them part of this essential isoprenoid biosynthesis pathway that I had explained earlier. So we showed this, and in a knockout experiment that I did with a colleague here, we also showed that if we knock out the SUF-S gene, the cysteine desulfurase, we do not see any sporozoites in the salivary glands of the mosquito, which essentially tells us that this is also essential in the mosquito stages of the malaria parasite. So you knock out SUF-S, you do not see any sporozoites in the salivary glands, and you do not see sporozoites in the salivary glands because they are not able to develop in the oocysts. This is the wild type parasite, which shows normal development of sporozoites in the oocysts, in the midgut of the mosquito. But here you do not see any sporozoid formation in the knockout. And this knockout of SUF-S completely inhibits sporozoid maturity and formation, and therefore tells us that this pathway is essential not only in the blood stages, but also in the mosquito stages, and therefore is really important. And if we can design an inhibitor or look for an inhibitor for this pathway, we would be able to have a multi-stage drug. Now, another pathway that we worked on, and I'll just show a couple of slides on this, was the ISC pathway for iron sulfur assembly in the mitochondria. This pathway is sort of conserved in the human, but we found from our experiments that it was also quite different from what we know in the human. Many of these genes have been, so in a large analysis of all Plasmodium genes using piggyback vector transposon mutagenesis, people have made these knockout lines, and it's been shown that many of these ISC pathway genes that are all nuclear encoded and are putatively mitochondrial transported are essential for parasite survival. And we expressed each of these and made antibodies against them, and then by immunolocalization experiments clearly showed that they were all mitochondrial. So they were nuclear encoded and went to the mitochondria. And we also established that the ISCS was a cysteine desulfurase whose activity was enhanced manifold by an interacting partner. We could in vitro assemble the entire iron sulfur complexation pathway. And this was really difficult to do. As many of you know, expressing malaria proteins is notoriously difficult. And that the fact that we were able to do this was in itself a big thing for us. So we could assemble the entire pathway. We could put this entire complex together and start seeing in vitro iron sulfur complex assembly. And what was interesting was that unlike all other known ISC pathways, what we assembled was a 4 iron 4 sulfur cluster, not what is usually assembled as a 2 iron 2 sulfur cluster. This was a unique phenomenon in the malaria parasite, not seen before in any of the ISC systems. And we confirmed this by exposing to oxygen, and we found that even if you oxidize this, you do not see a conversion to 2Fe2S. You just see a loss of 4Fe4S. So there was no 2Fe2S intermediate in formation of the 4 iron 4 sulfur complex. This is the scaffold protein on which the iron sulfur complex assembled. It's called PFICU. We did a access structure analysis of this, and we found that it's a trimer. And that you have, and by doing various measurements and actually measuring the number of iron and sulfur molecules, we understood that this trimer assembles a single iron sulfur complex. So either any one of these would carry an iron sulfur complex, or you would have an iron sulfur complex formed between these two subunits. Each of these subunits of the ISCU can, in principle, hold an iron sulfur complex here, but we still do not understand whether this happens or whether two subunits come together to hold a single 4s4 iron sulfur complex. This is something that needs to be worked out. Then because we had this whole complex assembled, we also tried to solve this axis by using small angle X-ray scattering to understand what this entire complex was like. And to our utter surprise, we found this normally in other ISC systems, such as in yeast mitochondria or in bacteria, you have two surface molecules forming a dimer interacting with two other U molecules and an ISD11 each. But in the Plasmodium complex, you had a further dimerization of this entire complex. So you actually had a heterohexameric structure which was further dimerized. And this is the kind of formation that you saw. This is where you see this here. And why was this dimerized? It was dimerized because in the surface protein of the malaria parasite, you have a large low complexity region at the N-terminus. And this low complexity region has repeat elements. It's very disordered. And it turns out that this is the interface between these two independent dimer units. And the interface is here. It forms a number of covalent and number of electrostatic and hydrophobic interactions. And this segment is missing from all other ISCS proteins known. And because it's missing, it's understandable that this sort of dimerization does not happen in other systems. So the complex is different, it's larger, it's further dimerized, and the ISCU protein only makes a 4 iron 4 sulfur cluster directly and does not make an intermediary 2 iron 2 sulfur cluster. We also identified the proteins that would help in transferring the iron sulfur cluster from the scaffold protein onto the target proteins. And we showed very specifically again that this ISCU, which assembles a 4Fe4S cluster, can use a carrier protein, which in our case is ISCA2 from our experiments that I show here, but can also directly transfer to a target protein. So it does not require a carrier. It can do both. It can work via a carrier protein, but it can also directly transfer a protein, transfer a 4 iron 4 sulfur cluster onto a target protein. So this is the final analysis where we understand that that the ISC pathway is conserved only partly, as if we compare it to yeast pathways and to the human pathway, that there are many differences. The major difference being that it does not assemble a 2Fe2S cluster, it assembles a 4Fe4S cluster directly, and that it has as much very different stoichiometry. It is further dimerized because of this unique extension in the ISCS. And in addition to that, we worked out many other complexities, but I'm not describing them today. So my last slide then is just to say that iron sulfur cluster assembly is an essential phenomenon in an organism, and that if you knock out iron sulfur cluster assembly pathways or inhibit them, it would cause parasite death. For mitochondria, it is actually believed to contribute to the essentiality of mitochondria for life. And so if you look at organisms which have lost mitochondria or have very reduced mitochondria, for instance, organisms with mitosomes or hydrogenosomes that have lost typical functions like the citric acid cycle or the respiratory and the heme synthesis pathways, they continue to retain FES biogenesis by the IC pathway. So this is the mechanism because of which mitochondria are essential for life. And very recently, just last month, a paper for toxoplasma has confirmed our results and shown that if you knock out the SUF pathway, even in Toxoplasma gondii, which is also a very important disease pathogen epicomplex in parasite, it leads to the loss of epicoplast and it leads to parasite death. So we believe now that it's really important that we start looking for drugs for this pathway because this is essential for parasite survival. And this is unique to Plasmodium. It's not found in the human. It's in the epicoplast. And this offers a very interesting avenue for drug discovery. I will end by thanking my students, Kirti and Ankit, who worked on the mitochondrial ribosome biogenesis and the KSGA functions, Vijay, Manish, and Sadik, who have worked on the iron-sulfur complex pathways, and my current lab. I just want to show that we are very gender-balanced historically. All the funding agencies and my collaborators. Thank you.
Thank you. Thank you very much, Dr. Saman. I think excellent talk. I mean, it's a pleasure to listen to you. I think after a long time, I listened to you. I was following your work. See, before I open to the other people in the audience, my question is, for example, these all advancements, whether they're in toxoplasma or in the plasmodium, which you talked about, can they make a difference to the therapy in the short run? Yes. Long run, yes, they have. But in the short run, choosing something which is already available and fitting in there for inhibiting these pathways. Yes.
Well, sir, both strategies have to be adopted. I mean, it's much faster if you use something that's known, right? But if you look at the way malaria drug discovery is moving today, internationally, people are working on new pathways and new proteins. I mean, people have stopped working on really artemisinins and so on and so forth. What is really happening now is ATPase4, ATP4, PFATP4, ATPases, DHODs. These are all new targets being looked at. So people have worked on them. They've identified these targets and immediately drug development has started. So I think people are thinking differently today because we now understand that we should not work at the older molecules because of resistance issues. And I think it's good to look at newer targets and work on them fast. So yes, both strategies have to be adopted. We still have to work on modifications, say, of quinolines and et cetera, but we should look at newer targets and work faster in these domains. I mean, a very good example of this is the way the phosphoromycin trial started. Hassan Joma was the person who worked out the essentiality of the isopenoid biosynthesis pathway in the malaria parasite Epicoplast and immediately started working on the inhibitors. And they've got clindamycin, phosphoromycin combination in phase 3 trials today. So I think it's important. So these were biologists who pushed it so that they got into phase 3 with a new target.
Thank you, Saman. Any other questions, comments on this?
Summer recently they have
Dr.
found
Kamboj,
gamma,
yes.
Summer recently they have found gamma and delta cells in the immune cells which are involved in protection. Any comments on this?
So I wouldn't, it's not something that I'm confident to answer on because I've not worked on this at all. But yes, I mean, the immune evasion strategies of the malaria parasite are notorious, as I said earlier, because it has this multiple ways of countering immune responses. There's of course antigenic variation, there's antigenic diversity. And I think at both these levels, and the complexity of its multiple stages. Also, in falciparum, there's also this additional complication of this low complexity regions, these LCRs, which are large, these are up to 140 amino acid regions, which are low complexity. And this mask epitopes. So they actually have an epitope masking system. So it's actually very difficult to work on vaccines. And one understands why it's so difficult to get a malaria vaccine. It's heavily evolved in terms of immune evasion.
Thank you.
Is there any possibility of seeing a anti-malaria vaccine in coming 10 years or so?
I mean, there is, one hopes.
There's already a vaccine,
yes.
So there's a report that I read in the newspaper that someone has come out with a good one. I'm not.
The latest, sir, is this RTSS vaccine, which is a CSP vaccine with an adjuvant. and it's that's one that's been cleared by the WHO for use in Africa.
Okay.
The efficacy, the protective efficacy of this vaccine is quite low. It's about 30 to 40%, but it's used in children. So the idea is that if you use it, so it won't come to India for sure, because the idea is that if you use it in a highly endemic, high transmission zone, then you can actually lower the transmission of a particular disease and therefore lead to protection of a large number of children.
Okay.
So I think that's where it will be used. And the rationale is that even if it does not have very high protective efficacy, it might drastically reduce transmission, which in itself will be very useful to, for children, especially to reduce disease.
Okay.
Someone, they say now there is another one which is 70% protective that has gone to multi-center trial now in Africa only.
Yes, sir, it's in trials now, but only when,
Right.
when the RTSS was being tested, also the expectations were very high. But it's only when the trials get completed, sir, will we actually get a picture of what the real protection is. So we hope that something will come.
Well, Dr. Saman, I think I'm not seeing any other hands. So must thank you for
very
a
much, sir. Thank you.
wonderful talk actually, which will stimulate many people, because the malaria is going to remain a trouble, with us for I think ages, in a different forms, different, maybe small number, but still killing, still being a cause of morbidity. So I think whatever you are doing, I think that should lead to a better and better therapies, because they will be needed actually. So I'm not never thinking of eradicating it, because it will never happen. So realistically, science is relevant. Your science is very relevant actually. Wish you good luck.
Thank you,
Thank
sir.
you very much.
Thank you.
So much of you. Thank you, Brahmachari Saab. Nice seeing you today actually after a long time. Yes. Yes. Thanks everyone. I think very nice session. I think which has given us a lot of food for thought, because as I said, we a limited number of people participating, but all of us really are amplifiers. We amplify what I think we can amplify to so many people. So I think that's our role. I think we have excellent session from that point of view. Thank you, sir. Thank you, Kambo Saab. Thanks, Dr. Saab. Everybody.
Thank you.
Dr. Chandrima also, if she's still around here. Thank you very much.
Habib, seeing you after a long time.
Thank you, sir.